para mí presentar a la doctora María Laura Gennaro. Ella es investigador principal del Instituto de Salud Pública de la Escuela de Medicina de New Jersey, de la Universidad de Rutgers. Y es muy especial esta invitada porque fue mi jefa un par de años atrás, por no decir varios años, ¿cierto?
3
varios años
4,
atrás.
o 4.
Exactamente, 3 o 4 años solamente. Marila, los intereses de investigación de Marina están centrados en enfermedades infecciosas. Ella tiene por sobre 100 publicaciones en prestigiosas revistas científicas y hoy nos presentará sus últimas investigaciones porque ha estado trabajando en COVID con SARS-CoV-2. ¿Ok? Marila. ¿Ok, Marila? You can present.
So, good morning, everybody. Buenos dias, and I'm sorry I cannot give this talk in Spanish, but so I'll give it in English, and I kind of understand Spanish some, so if you want to ask questions in Spanish, I'll try to handle it. And so let me get started. Let me just share my screen. Do you see my screen?
Yes.
Okay. So the title of my talk is the blood landscape of COVID-19. And before starting, I wanna thank all the organizers and Tali in particular. Italy has a special place in my, heart and in my career because she was my very first trainee. So, it was the first person on which I tried to be, somebody's boss, and I couldn't have found a better person. And we became friends, and it's really a joy to be here today. So before I start, I want to first thank the people that did the work, Pratik, Natalie, Rahul, and Pankaj. And it's not just a normal acknowledgement slide. These are people that worked without taking a day off during the entire pandemic, and they've been not only excellent scientists, but they've really been, making personal sacrifices to be able to do their work. So I think they deserve not only a scientific, but also a human recognition. And this slide is mostly, these are people that, that you don't know, but these are all people at Rutgers. There are physicians, basic scientists, physicians of all kinds. And I made this slide to show how much interaction, how much collaboration during the pandemic has developed among different people many of them I didn't even know before the pandemic started. So if there is silver lining to the pandemic has been to start working with other people and kind of seeing things that perhaps before they weren't so obvious to us. So first I want to, as I said, the title of my talk is the blood landscape of COVID-19. And so the first question to ask is, why do we talk about blood in a respiratory infection? So as you know, the SARS-CoV-2 is a respiratory virus. It infects epithelial cells in the respiratory tract, and the epithelial cells that when they get infected, they start releasing some danger signals first to the neighboring cells, other epithelial cells, endothelial cells, alveolar macrophages, and all these cells that become aware of the presence of a pathogen, then become making their own signal molecules. And so these these are mediators of inflammation. They go into the circulation, and they start recruiting to the site of circulation immune cells of various kinds with the goal of achieving, in the case of a beneficial or positive outcome, achieving on one hand the neutralization or the blocking of the ability of the virus to infect, or the clearance of infected cells. However, in some cases, inflammation becomes excessive, and when there is hyperinflammation, then it means that multiple cells, both resident and that have migrated to the site of inflammation, start releasing release additional cytokines, and this cytokine storm has been causes damage to the lung, and then through the circulation it can reach other organs, causing multi-organ damage. So the blood is important because essentially is a highway through which immune mediators and immune cells go throughout the body, and they mediate those events that they either contribute to a beneficial outcome or to a harmful outcome, and the disease become worse or fatal. And in addition, the blood is, as we all know, it's an accessible site, so it's a tissue that can be harvested much more easily than others, and so that makes also a big advantage. So I'm going to tell you about 3 studies that we have been doing in the lab, and let me start with the first one. The first is about seroepidemiology. Actually, this is why we started the whole lab started with this idea. And as you know, we've all become, even non-scientists, everybody has become an expert on how to diagnose COVID-19. I found myself talking to my sister who has a PhD in French, talking about IgG and IgM, and I was saying, I'm talking to my sister about immunoglobulins. So, so life is interesting. So we all know that, upon infection, the viral load increases, and this is, detected by a PCR. And at that time, still there is no antibody response. Antibody response starts with some delay relative to the infection. So it decreases with some delay, but the signal, so the antibody in circulation, remain for a longer time than the swab is positive, or the nasal nasopharyngeal swab is positive for PCR. So the PCR result is more short-lived as a result than the antibody signal. So if one thinks of, wanting to know what is the true burden of the pandemic in whatever area, that can be as small as a town, as big as a continent, and let's say that the true burden is the iceberg, is the entire iceberg. With PCR, one can see only one part of the iceberg, and at the same size of the iceberg, one can say by PCR that there is this many or this few cases or this many more cases, and that depends on when the people are tested, given the fact that it's a much more transient signal, or as it was, for example, here in the US at the beginning, there was little access to PCR testing, so many people didn't get tested, etc. So the PCR is not a good tool for epidemiological purposes. So we used antibodies for the reasons that, because as I said, it's a much more stable marker. And in our, to do antibodies, one usually uses, there are various ways, but one uses an ELISA. Again, we all know what the spike protein is. It's a glycoprotein that is that decorates the surface of SARS-CoV-2. And we chose to detect antibodies against the portion of the protein that is called the receptor binding domain. That is the domain that binds, that interacts with the host cell. So it binds a receptor on the host cell. And we chose it because A, is immunodominant, and B, because among the immunodominant antigens produced by the virus is the one that has the least, shares the least amount of sequence with other coronaviruses. So it's the most specific portion of the, among the immunodominant antigens. And this can be seen well here. These are ELISA results. Each circle is a person, and these are IgG titers against RBD, and you can see that it's very easy to distinguish those that have been COVID infected from pre-COVID sera or from people that they were not infected with COVID during the pandemic. So it's a good tool for seroepidemiology. So this is, also I want to show the lab to kind of brag about the fact that when the pandemic started, this lab did not exist. And as I always say, we built the plane while we were flying. So we started, pipetting, borrowing multi-channel pipettes from labs that were, empty because people weren't working. And then we ended with, thanks to the chancellor's generosity, we ended with all the automated equipment that we needed to do ELISA and other immunological assays. So what we did, so the seroepidemiological study that we did, it was centered in Essex County, that is the county of New Jersey where our campus is located. And we did the sampling based on people that were going to buy food. So they were, we recruited in front of grocery stores, supermarkets, bodegas, small and big, with the idea that even during a pandemic, everybody needs to eat. So somebody had to go and buy food. And that's kind of a way to do the least possible bias, to do a sampling of the population that is as little biased as possible. We had the mobile vans that would go in front of the stores and approach people, interview them, recruit, and if they were willing, they would donate blood by finger prick. There was then, you do a finger prick, then this little microsampler sucks up 20 to 30 microliters of blood. Then this can stay at room temperature actually for a long time. Comes to the lab, this blood that is there is eluted, and then one can do all the antibody work, both for binding and also for effector function. And actually many other plasma markers in principle can be studied from there. So the results of this study were that, as expected from what I told you in the first slide, or in the slide on PCR versus antibody, we ended up seeing that the burden of infection as determined by serology was more than 6 times larger than what the PCR testing, self-reported PCR testing would have indicated. And also, and I think now has become quite, common knowledge, that the number of people that reported among the seropositives, the number of people that reported no symptoms, that had no symptoms, were actually somewhat bigger than the number of people that reported having had COVID or COVID, or symptoms consistent with COVID. So that's one study. The other study that we've done, we started, looking into, the properties of convalescent plasma, especially at the beginning of the pandemic here in the US. That started in March, April of last year. It had become an important therapeutic tool. And the reason is that to this day, these two are the only drugs that are actually used routinely for COVID patients when they are in the hospital, an antiviral and a corticosteroid. So especially during the height of the pandemic, a lot of attention turned to use of convalescent plasma, because of course the convalescent plasma contains antibodies that in principle by passive transfer could help the recipient, a COVID patient, recover and block infection and recover. So the approach that we took was to study in parallel both the plasma of a donor that had been infused into a recipient and see what the outcome of that transfusion had been. So it's in its own a particular way, it's in reality is an in vivo assay for the efficacy of the donor, of the convalescent plasma, donated by for passive immune therapy. And so I'm going to tell you about the first case history that we had. So this is the recipient. The recipient is a woman, a female, Caucasian, aged 52, with an underlying autoimmune disease. She was infected very early in the pandemic in March of last year. She was hospitalized twice, and she had all the symptoms and also the signs of left lung pneumonia. She was positive to SARS-CoV-2 by PCR, and despite whatever treatment was done, this is now more than a year ago, she spiked fever twice a day, and she had the first infusion of convalescent plasma, and there was no improvement, and the fever continued to spike twice a day. So this was brought to our attention because they wanted to know what was going on with the patient, and also whether in her family the disease, husband and an adult son, the entire family had been infected. So we tested for antibodies, the family, and while husband and son had very high levels of antibodies to RBD, the patient didn't. Essentially, this is kind of baseline. So she had no detectable antibodies. So she was transfused with her husband. The husband happened to be compatible. She was transfused with his plasma, and within few days, the viral load dropped dramatically. In the the nasopharyngeal swab, the fever disappeared. She became PCR negative or with really low CT numbers. She received the second infusion, after which she became PCR negative, and she was released from the hospital. And as far as we know, she's been fine since. So the first conclusion of this work is that husbands can be useful sometimes. So, these are the blood draws that we took from the patient. The two darker circles are at the time of the two transfusions, and so we had blood taken before or at transfusion, and then for a period until she was discharged from the hospital. So first we looked at antibodies. To viral specific antibodies, and we saw that upon transfusion, of course, her titers increased. They decreased a bit. With the second infusion, they increased again, but they remain stable, at least for the duration of hospitalization. Interestingly, we looked at the presence of viral RNA in plasma by PCR, and we found, and we used two sets of probes and primers, one against nucleocapsid and the other one against RNA polymerase. And we found that in coincidence, or right after the drop of the viral load in the respiratory swab, these are the two vertical bars are the two transfusions. So after the first transfusion and then after the second, this signal of viral RNA in plasma decreased very drastically until it completely disappeared. Interestingly, the signal for RNA polymerase was never detected. So that indicates that this signal here, the nucleocapsid signal, is not from genomic nucleic acid. Otherwise, if it was the genome of the virus, we would have a similar signal from both genes for obvious reasons. So the fact that we see only nucleocapsid and not RNA polymerase it suggests to us that this is not genomic RNA, but that these are viral transcripts from infected cells that probably are lysed during the reaction that is caused by the therapy, and so release the viral transcripts, and that's consistent with the fact that the nucleocapsid transcript is much more abundant in infected cells than the RNA polymerase transcript, because there is much more nucleocapsid that needs to be made. Since nucleocapsid is a structural protein, then you need enzyme so the copies are much, in a much bigger number for the nucleocapsid. That's what we saw. We also looked at cytokines in plasma. So this is a heat map. We did, I think it's about 40 cytokines. Each column is a cytokine, and each row is a day of of the blood draw, and it goes, the transfusion, the temporal goes from the bottom up. So day 0 is the time of the first transfusion, and then it goes up. And basically, when the data were analyzed by, unsupervised clustering, then we found that there were 4 clusters that formed, and these 4 clusters are temporally aligned. So they're, they go into, so there is the kind of one cycle after another of something that is happening in the patient. And a very similar result was obtained when we looked at the plasma proteome. So this is now plasma proteins is exactly like the one before, except there are many more vertical lines because there are many more proteins that were tested, and we found the same thing. 4 clusters, they were, temporally ordered, very similar to the cytokine picture. We also did a single cell transcriptomics of her peripheral blood, and we saw that the most noticeable difference was in the monocytes. If you can see over time, these are 3 samples. This is the signal that changes the most. Over time, the others are more or less the same. And so these are innate immune cells, and the reason underlying this graphical difference is the change in innate immune responses to the virus, like type interferons and various other inflammatory markers. So going now on the side of the donor, we characterize also the convalescent plasma that the patient received. And as I said before, the donor, the husband, had a high titer of IgG. This is a titration curve, and most of the IgG were of the IgG1 subclass. And then we also asked, so we set up a neutralization assay using live virus. Since we are tuberculosis investigators, we are very conversant with BSL-3, so we didn't find very difficult to set up cultures with live stars in our BSL-3 lab. And so we asked the question of what are the antibodies that are responsible for the neutralizing activity. To do that, we took the plasma, prior to the neutralization assay, we pre-absorbed it with either nucleocapsid or with RBD to deplete it from the plasma from the specific antibodies. And these are ELISA data. They show that if this is the signal before pre-absorption, when you pre-absorb with nucleocapsid, the signal to RBD is not changed, but the signal to nucleocapsid is much lower because you have depleted the plasma from anti-nucleocapsid antibodies. And the same thing, happens if you do the RBD pre-absorption. And what this right panel shows is that prior to pre-treatment, the plasma was responsible for about 80% inhibition of viral replication, and that ability to neutralize the virus was severely impaired if the plasma was pre-absorbed with RBD, but not if the plasma was pre-absorbed with nucleocapsid, indicating that the viral inhibition is associated with antibodies to RBD rather than to antibodies to nucleocapsid. Okay, so this is a summary of what I said. The decreased viral RNA in the plasma of the recipient probably derived from lysed infected cells. We've seen this in other recipients of transfusion that in a study that is ongoing, and this transition, as I said, across multiple states that is defined by this changing in the plasma protein, also in the gene expression patterns of the immune cells. And on the donor side, we found that the donor plasma had high levels of RBD, IgG, mostly IgG1, and that the neutralizing activity was mostly RBD specific. So time passes, so we go from studying, in this third study, from studying only infection to also studying responses to vaccination. And when we started this work, these are the questions that remain still the kind of the hot questions of the day. If a person is infected, how long does protective immunity last? What is the interaction between vaccine and previous infection? How does infection affect the response to vaccination and vice versa? Do the responses to vaccination and infection change? And of course, how long does protective immunity conferred by vaccination last? We haven't at all addressed this. I think it's too early, at least with the type of people that we have at our disposal. So we tried to address the first three. So this is the study design. So early in the pandemic here, around April or so, we started recruiting people here, Rutgers employees who had had COVID and had recovered. And we collected blood samples for about 8 months. At this time, we thought that the study was completed, but vaccination started, and so some of this group got vaccinated. So since we had the pre-vaccination samples, we had the opportunity to collect also the post-vaccination, post-vaccination sample from the patients, and in order to or the subjects rather, in order to understand the response of people who had been infected to the vaccine, we had also a group of people that were not infected, so they were infection naive, and also they had gotten vaccinated. And the kind of assays that we did are the ones that I showed you earlier, antigen binding, viral neutralization, and we also looked at, for reasons that I will explain in a minute, at B cells and specifically at virus-specific B cells. This is the cohort. What I want to pinpoint is the fact that the vast majority of the subjects that we recruited had not been hospitalized for COVID, so they had had mild to moderate symptoms. And of these few, actually, they were hospitalized. None of them belonged to the subset that had received the vaccination and then came back to us. So they were not part of the post-vaccination work. So only mild and moderate cases. So this is, I showed you earlier this. So first, there's all, essentially most, not all, but most of the subjects had antibodies to RBD. And as we followed over time, we saw each line is a subject that in the vast majority of the subject showed a decrease in antibody levels over time. A few remain stable and 2 or 3 increased, perhaps due to reinfection. We don't know that. Very similar, we obtained very similar results with neutralization. Neutralization titers decreased over time with a few exceptions. And again, the neutralization activity was associated with RBD specificity rather than to nucleocapsid, doing the same kind of pre-absorption experiment that I described a minute ago. So they decrease with time, they correlate with the anti-RBD antibodies, and they correlate because the neutralizing activity mostly resides in the RBD specificity. So then, of course, this is the time where you would see on social media, on newspapers, oh my God, antibodies decrease, we're not protecting, infection doesn't protect, and even, how can I say, valued or, people that should know better, they start, freaking out about this. And honestly, it's incomprehensible, because if you open a textbook of immunology, the first figure that you see is that when you have an IgG response, that declines over time. And that's physiological. You cannot maintain, unless you are encounter antigen again, you cannot maintain a high level of antibodies. If you maintain a high level of antibodies without encountering antigen, you have an immune disease. A normal person has this physiological decay of antibodies. So the question was, okay, antibodies are decaying, but does immunity persist? And the immunity does not, the memory of the immune memory resides with the cells, not with the circulating antibodies. So we started looking at B cells. So here we have in the top panel, we have the gating strategy, and essentially, you know, the B cells, we define them as being CD19 positive, CD20 positive. Then we separated with CD38 and CD27, we separated the non-plasma blast from the plasma blast, and the non-plasma blast can be further divided by IgD and CD27 expression into 4 subsets of memory B cells: naive, non-switched memory, switched memory, and double negative memory. We also used an RBD tetramer that binds to the B cells, and so we could actually assess the frequency of RBD specific memory B cells in the subjects. And we found a transit. So here these are you know, the results of the flow cytometry. The blue are the people that were never infected, that we used as a kind of a reference point. And then these are the same subjects around, you know, at the first blood draw, about, you know, within 2 months since their symptoms, and at a later time, 5 to 8 months later. And we saw a transient increase in plasma blast in the infected people. This is again, it's actually something that has been in many acute viral infections, so there is nothing new about it. And as expected, we saw that relative to people that were not infected, the infected people had an increased frequency of virus-specific B cells, and importantly, the virus-specific memory B cells do not decrease over time. So even though the antibody titers decrease, the pool of virus-specific memory B cells remained unaltered, at least for this period of time. We were kind of intrigued by the fact that this particular subset, the double negative, that means that they're missing both the memory marker CD27 and IgD, that is lost as the B cells acquire the specificity, say, for IgM, IgG, IgA, etc. So these double negative cells, which I'll tell you a bit more in a minute, increased over time in the infected people. And what is interesting is that what we noted is that they increased post-acutely. So they were found increased. These are the same subjects. Green and red are the same subjects. They were increased only at the late time point. And this happened with the RBD double negative B cells, but when we looked at the total pool of double negative B cells, we saw the same phenomenon, that even the total pool of double negative B cells increases post-acutely in people that were infected, even though the infection was mild. But so that's what the finding. It became even more interesting when we looked at the results of the effect of vaccination. So just focus on these panels here. So here we're looking at people that were not infected, so, you know, non-infected or infected, and before and after vaccination. So this group is non-infected and vaccinated. These are infected, not vaccinated, and then the same people infected after vaccination. After vaccination, and what we saw is two things. One is that everybody, regardless of whether or not they'd been previously infected, had a strong response to the vaccine. The fact that this is less than this doesn't mean that it's not strong. The titers actually are very high. But that the subjects that had been previously infected had a much more vigorous response in terms of both antibody binding, neutralizing titers, and virus-specific B cells than the people that were not, they were infection naive, that had never been infected before. They'd never been infected. So that's number one. The second interesting observation, going back to the double negative B cells, was that, as I told you before, there was an increase we saw an increase, post-acute increase in these double negative B cells in infected people. After vaccination, and again, I stress, this is the same people, the levels of double negative memory B cell decreased to baseline levels. So vaccination countered this expansion of double negative B cells that was induced post-acutely by infection. So why is this? So then we started thinking, you know, what's happening here? What are these cells? So let me tell you in 1 minute. This is B cell immunology in one minute. So this is a secondary lymphoid organ. This is a follicle, and you know, in the follicle there is the B cell zone and the T cell zone. And in a normal situation, when there is an antigen, it could be an acute infection or something else around. Both the B cells and the T cell experience the antigen, and when they become antigen experienced, they meet at the periphery of the B cell follicle. Everybody stays in their area, and upon interaction with the T cell, the B cell pool undergoes an expansion and enters the germinal center. The B cells that enter in the germinal center, they undergo two very important phases of development. One is somatic hypermutation, and the other is affinity maturation. And this leads to the fact that both the plasmablast and memory B cells become capable of producing a high affinity antibodies. So the plasmablast produce antibodies, of course, and the memory B cells have the potential to become plasmablasts that produce high affinity antibodies. Physiologically, a smaller proportion, this is why this arrow is thick and this is thin, a smaller proportion of cells, of B cells, never enter the germinal center, remain extrafollicular, and they give rise to plasma cells that express low affinity antibodies, or to clones. So since they have not gone through the germinal center selection, for example, the autoreactive clones have not been deleted, so they in principle can also produce autoantibodies, because the autoreactive clones haven't gone through the elimination in the germinal center. What happens in some conditions, mostly in chronic infections such as HIV, malaria, and there are some other infections that show this, or in autoimmune diseases, is that after for reasons that I'm not going to go into, after the B cell expand, instead of going into the germinal center, they predominantly remain extracellular. So as a result, you have more cells that are either dysfunctional, or they produce low affinity antibodies, or they produce autoantibodies because they have not gone through the germinal center selection. And this is what happens, for example, in HIV, this double negative. So because it's a heterogeneous population, they are called double negative, extrafollicular, atypical. They're not exactly the same because they change from disease to disease, but more or less we're talking about the same cells. And these are cells that in HIV have been described as prematurely senescent or exhausted, and perhaps they are linked associated to the fact that there can be poor antibody responses in HIV infected people, or in autoimmune diseases such as lupus. They have been associated with production of autoantibodies. So these are cells that are either dysfunctional or pathogenic because they produce autoantibodies. So that's what we saw in terms of COVID that are now as long COVID. The question arises, and that's a question that we're asking, is whether the double negative B cells are at least one of the reasons why some people have persisting symptoms, and whether the effect that the vaccination has on these cells could be one of the reasons why the vaccine seems to be having a positive effect and in relief of long COVID. So that's the end of my talk. The conclusion is get vaccinated, whether or not you were infected with COVID. And I'm here, I thank you very much for your attention, and I'm here to take your questions. Thank you.
Thank you very much, Marila. It was a really interesting presentation and updating us with the response of the vaccines. I don't know if somebody from the people who's here would like to present something, would like to answer questions to Marila. I have a question, Marila. You mentioned that everybody is producing antibody, right? After the vaccination.
Well, everybody, I cannot say everybody produces antibodies. No, for example, there are people that are immune suppressed or they're taking immune suppressive therapy. They may not produce antibodies, unfortunately. Yes.
But there is not a baseline of what amount of antibody is better for being protected or not, because it depends on each organism, right?
Yeah, I mean, first of all, the antibody level doesn't necessarily reflect the function, right? So if, for example, what we've shown, and many other people have shown at this point, is that the antibodies to RBD correlate well with neutralizing power of the plasma for the reasons that I described. And therefore, there is a good sense, if you have more anti-RBD antibody than me, most likely your plasma has more neutralizing power than mine.
Yes, yes.
But you cannot, but what is enough? And that also depends on I think, I mean, I have no proof for that, but I think also depends on infecting dose. It depends on, how much, what is the dose of infection that you get, how well you can, it's possible that even if you have antibodies, if you are exposed to a high dose of virus, it may not be as easy to neutralize it as so no. I don't know. I don't think there is no magic number.
Yeah, there is no magic number between organisms, because they function differently every time, and it depends, as you say, the load of virus that you have in your disease. Okay.
It could.
It could be. Because here, since we are in winter time, we are also vaccinated against influenza. So I will talk about myself. I got vaccinated the second dose of Sinovac at the beginning of April. And on May, I vaccinated against influenza.
Mm-hmm.
This will move all my my physiological function of antibody, or?
Oh, I don't. I mean, I think that is. I think it's fine. I mean, it's. I mean, in principle, each vaccine kind of goes to talk to its own subsets of B cells. There shouldn't be a lot of interference. There should not be a problem. I don't think that any problem is, at least as far as I know, has been reported in one preventing, efficacy of another vaccine, one vaccine. I mean, we give to children a bunch of vaccines all at the same time, right?
Yeah.
So,
you're right. Yeah.
You're
right. It shouldn't be a problem. I don't know if somebody else would like to ask something to Marila. I have people who raise their hands.
Yeah, I saw that in the chat, somebody said, I have a question.
Yeah.
Christian or Anna?
A long one. Okay. Is Christian? Would you like to ask Marila?
Yeah, I would like to say something.
Sure. Just go ahead.
I was wondering if there is any difference between antibody counts between, for example, Pfizer vaccine and the Sinovac vaccine, or any difference between RNA and viral load vaccines.
I don't know. I don't know. We haven't we haven't done anything like that. I mean, the people that we had recruited that had either, I mean, mRNA vaccines, either one or the other. But here, because these are the vaccines that we had here, and then the Johnson & Johnson vaccine came along at a later time. So when we recruited these subjects, there were only these two. So no, we have not. We don't have access to people vaccinated with other vaccines. I mean, for example, most of my family in Italy has been vaccinated with the ones that have been vaccinated with AstraZeneca, mostly. So I'm sure that there are studies, or there may be studies, but I'm not aware of those studies. I'm pretty sure that people have compared. But what I tell whenever somebody asks me, people, family, as you can imagine, family has called me from Italy and etc. I say the best vaccine is the first one you can get.
Yeah, exactly.
That's my scientific and non-scientific recommendation. The one that you can get is the best.
Yeah.
Yeah, grazie mille.
Grazie mille.
Prego.
Gracias, Christian. I have a lot of students here from my course in molecular biology. Somebody else would like to ask something? If
somebody wants to ask in Spanish, please go ahead.
Yes.
We can handle.
They are shy, I guess. Okay, I guess nobody else is gonna ask. I have another question for you, Marila. When you talk about the B cells, memory cells, the memory cells, you said so that first, I mean, they will remain active after the main receptor.
So the memory cells are, or some of the memory cells are, that's where their memory, right? They are tucked away and they stay there and then again, upon encounter with antigen, they will, be informed by the antigen presenting cells that there is antigen again, and that will stimulate what I, just showed, and they will, and of course, the second, if they see it for the second time, the process, as I showed you, is going to be much faster because you don't need to go through this whole process. The antigen-specific memory cells are already there. So how long? I'm always very curious when people say, "Oh, the vaccination of the last 6 months, or doesn't last more than 6 months. But if we have vaccinated only for 8 months, how do we know how much it lasts, right? Or how we know what we know. So we know that, so what I can tell you is that in the time frame that we tested those people, the memory B cells, the virus-specific memory B cells had not changed. Now, of course, with vaccination, there is going to be, a boost, a boost of those cells. So if people are vaccinated, they're going to have, more, and they're not going to go down. So, I don't know. In principle, one would have to follow people that have been infected, they don't get vaccinated, and they, and they potentially are not. I mean, it's not easy, because to really answer that question, you have to have somebody that is infected, that doesn't get vaccinated, and also doesn't get re-exposed. Yeah. And then you can, so you can monitor for as long as you can monitor. You can say what you can say, right? So that the thing is that with this pandemic, I don't know, even scientists sometimes they forgot their science. So if you can say something up to 8 months, how can you say that this does, how can you say? You have to say you don't know. It doesn't put you on a TV show if you say I don't know, but that's the truth.
Yeah.
Right?
Yeah, exactly. Yeah. If it is 6, 8, or a year, we don't know.
We know from, from day one. I mean, the earliest was in China in December or November of 19, right? That's as long as we can know.
Yes, yes.
So,
it's completely something new.
Yeah.
That is affecting us everywhere. Okay, Marila, I guess there is no more question. Nobody asked.
Okay.
So many thanks for your talk.
Somebody has another. I think Christian said he has another.
Yeah. Go ahead, Christian.
Yeah, I'm sorry. So my question mainly was about for example, there are several, not studies by itself, but for example, in social media, there's this thing about Pfizer, for example, or AstraZeneca, that in some people they create blood clots and due to the vaccinations. I'm not sure if that's a certain study, of there is anything relating that.
So what I can tell you is this, but this is now, it's not, I'm not talking as I'm talking as a scientist because I'm a scientist, but I'm not talking about work in my laboratory, what I think. So I think that with the AstraZeneca vaccine, I think that it's I don't have a doubt that there is a rare side effect that is this, effect on coagulation that you're referring to. It's been seen in different various countries, so I presume is real. The frequency, now I don't remember what the frequency of it is, but it's I think 1 in a million or something along those lines. So if you can, let me wear my, feminist hat, the birth control contraceptives cause coagulopathy in women 3 or 4 logs more frequently than the AstraZeneca vaccine. And I haven't seen, none of us has seen this public outrage about, birth control because nobody gives essentially a damn that, women take the pill and can get, So I think that there are 2 things. One is that everything has to be put in a context. And secondly is that every time you do something, you have to weigh your risk and benefits. As I have, a toxicologist friend, she says, not even water is devoid of side effect. So there is nothing that doesn't have side effects. The thing is that if something has this kind of side effects in 1 in a million, and the alternative is not to be vaccinated and risk what all of us unfortunately have seen with, in their countries, with their, what is how, what is the risk benefit? I think it goes, I mean, with no doubt, it goes in favor of getting vaccinated. That's my opinion.
Yeah, thank you so much. I agree with that, because the benefits outweigh the cons, the problems that they bring. So it's amazing actually to know that. Thank you so much.
Yeah, I know. The thing is that there are, with so many people, by the way, with mRNA vaccines, I don't think that there is a coagulopathy. I think even at those low levels. I mean, I think that we have, the fact is that we have vaccines that are, in an incredibly short time, vaccines have been given how can I say, the extent of the disaster that we've been, experiencing. In this short time, vaccines have been made that are incredibly efficacious and have very low, extremely low frequency of side effects. Some may have, for example, also the J&J vaccine has, has been reported with some of, this coagulopathy, again, at this low level. I have a daughter who's 26. She came from London. I had her vaccinated with J&J because she couldn't stay here for 2 doses. So I vaccinated my daughter with J&J. I mean, what else can I tell you? That's my best answer. I don't think that, and when my husband got all worked up about this, I said, did you ever ask your daughter whether she's taking the birth control pill, and have you ever wondered what is her risk to get a coagulopathy due to hormonal contraception? So just, of course, that doesn't deny the fact that that 1 million, one case in a million, it's a tragedy. But it's the frequency that is extremely low, and that I think we need to consider.
Yes.
Thank you so much.
Thanks, Christian. I have a raise hands iPhone. I don't know who is.
Sergio Marshall.
Sergio, hola. Sorry. Sergio, go ahead with your question. It wasn't a question, Sergio? No?
Also, is muted.
Yeah. So I guess that's it. Nobody else?
No, I'm here.
Okay.
I'm sorry. First of all, I would like to acknowledge you because it was a very nice talk. And what I would like to know, I would like to have your opinion that since herd immunity is not achieved, and we don't know when it's going to be achieved, is it possible to think that third doses of vaccines would help in order to protect more the population?
That's a good question. I think that, as you say, the estimates are they will not achieve herd immunity because we're not going to reach the numbers that epidemiologists have, or the people that know how to do these calculations have anticipated, achieving herd immunity. I think that, however, I think what I've read, but again, this is now not my science, is what I have learned by reading is that, and what I, what is an opinion, is not a fact, is that if even though we don't reach herd immunity, if we manage to, if we manage to contain the spread of the virus and therefore for example, as we all know, the rate at which these variants arise is proportional to the rate of replication, and the rate of replication of a virus is proportional to the number of people that it infects. So I think that what we can achieve most likely is what I hope we can achieve is to limit the circulation of the virus with vaccination, as well as other tools, because I think that we cannot rely only on vaccination. There are other tools that need to be implemented and are not implemented enough. For example, I think I'm a very strong advocate of antigen testing. I mean, the good antigen tests. I think that antigen testing is something that helps identify people that are infectious, and those are from a public health point of view, not from a medical. From a medical point of view, you want to know if your patient is infected or not. But as a public health official, you want to know if somebody spreads infection or not. So the antigen tests are good for that, and we should, I think that is a tool that is not used. In fact, it's used very insufficiently. Now I read that there are a few states in the United States that, for example, have started using them in the way that they should be cheap. And they should be, or of course, or be subsidized and therefore provided for free. And they should be outside of a school, outside of a stadium. Outside, I mean, we can go to the stadium and go see these damn, soccer games that we all like so much, and get antigen tested before we enter the stadium. It's as simple as that. Right. So if you limit transmission, you vaccinate people and increase the number of people that are protected. If you limit transmission by say identifying the people that are the spreaders, the spreader is a bad word because it sounds like somebody wants to do it on purpose. People that are infectious, right? And then you maintain some kind of behaviors. In some cases, I know that in many places masking is not very popular. Of course, it's not popular. Who likes to be masked? But if some of this is maintained, or in some circumstances, I think that altogether, plus you're in winter, we are in the summer, so everybody's outside. But obviously, respiratory viruses, get less. They spread less in outdoors and all that. So all these things together, I don't think that one can rely on vaccination only. Okay. The fact is that that everybody should be kind of a responsible citizen, and plus there should be these tools such as this antigen tests that they should be used much more than they are.
Okay. My last question would be, thinking that we have to optimize vaccines in the future for all the viruses that are showing up, do you think that messenger RNA is going to be the future for vaccines?
Now, this is really outside of my field of expertise. So my honest answer is, I don't know. I'm not in vaccine development, and I think I learned with the rest of the world about what it was already known about mRNA, as a vehicle for these vaccines. I don't see. I mean, what I read, but again, this is what I read, not what I know. Is that of course this is a platform that now. I mean, this has established an incredible precedent, and this can be applied, and I'm sure it will be applied. Is probably already being done for other vaccines, but that's really not my area.
Okay, thank you very much.
You're welcome.
Thanks, Sergio. Gracias. Well, I guess that's it, Marila. We are on time.
Okay.
Yes. Thank you very much. It was a really pleasure to have you here.
Same here.
So we can do it more frequently to have you in a virtual seminar, or well, when the time comes, you will come here.
In vivo, yeah. Okay.
Vivo, yes,
okay.
Yes.
Okay, in vivo. I've never been to Chile.
We'd love to have you here in vivo.
Yeah. Okay.
Thank you very much.
Thank you, and stay all safe, okay?
Stay safe, Marina. Thank you very much.
Bye-bye.
Bye-bye.